Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Stefan from Business Review, and I will be your host. It is our pleasure to have LTS AG with us today, who will be presenting this webinar titled Microarray Patch System. Today's guest speakers are Casey Sharp and Dr. Sebastian Braun. Before we begin, I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based. So if you disconnect for any reason, please just click on the link received via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. Just type them into the box and click submit. We will allocate some time at the end of today's session to address any questions or thoughts that you may have. Please use the yellow help widget if you require any assistance, and keep in mind you can move, resize, and maximize any of the windows in front of you for a better view of the slides. But now, please allow me to introduce Casey Sharp.
Yes, thank you, Stefan. My name is Casey Sharp. I head up the business development efforts for LTS here in the United States. My background is in chemistry as well as in business. I've been with the organization nearly a decade, and I'm happy to host you today in this webinar. Sebastian, would you like to introduce yourself?
Yes, thank you, Casey. My name is Sebastian Braun. I'm a molecular biologist by training. I'm in the field of dermal and transdermal drug delivery for 15 years now. I'm always in the drug development and manufacturing area. And I'm happy to welcome you today to give you a technology update.
Great, thanks.
Thanks, Sebastian. Handing over to
So, allow me to give you a bit of background about LTS, who we are from a global perspective, how we make money, how we do business. Traditionally, our background has been in coating technologies. From that coating technology, we derive oral thin films as well as traditional transdermal patches. And from that, we saw a unique need in the marketplace as well as in the patient population to be able to bring large and small molecules through the skin, alternative to the traditional passive transdermal systems. So over a decade ago, via organic growth, we looked into developing our own microarray patches, or microneedles as they're commonly referred to. We've developed and refined what we believe is a patient-friendly and a commercially scalable process to do that, and that will be the focus today. A bit of background of LTS from a global perspective. We have around 1,000 employees with sites in West Caldwell, New Jersey, which is mainly focused on manufacturing. We have our site in Annenau, Germany, which is our center of excellence for research and development as well as commercial manufacturing. And we have a business office in Shanghai, China, with thoughts and plans of having brick and mortar manufacturing capabilities in the Asian markets as well. We do roughly 300 million euros a year in sales, so quite a healthy company in the CDMO space. Of that revenue stream, we reinvest over 8% of that revenue back into the R&D organization, so roughly $25 million, euros a year, reinvested into our R&D efforts. We position ourselves as a technology company, and within that, we are cognizant of the need to build intellectual property around our technologies, as well as bringing intellectual property to our partners and their molecules and ultimately their drug product. So we have over 5,000 patents filed and over 300 patent families. So very important as part of the drug development process is building in intellectual property around our technologies. From a traditional sense, which would be our transdermals and oral thin films, we produce about 750 million units annually to supply the global market. I think that's important as in the future looking into what's really considered a novel technology. We have the experience to supply the global pharmaceutical market with drug products. So as a partner in the CDMO space, LTS checks those boxes and fits nicely within collaboration opportunities. To date, we have commercialized roughly 30 products with our partners, and that's LTS in a nutshell. Stefan, I send it back to you.
Thank you very much. It's now time for our poll question. Do you have previous experience of microarray patch microneedle technology? Yes, I have been dealing with the topic. Yes, but I have only basic knowledge. No, not at all. If the audience can please take the time and answer, you should see the poll right in front of you displayed on the screen. And now we can take a look at the results. So it's actually even 37.5% of people voted yes, but I have only basic knowledge, and the exact same amount of people voted no, not at all. And 25% of the audience voted yes, I have been dealing with the topic. Now on to our next poll question. Which of the following best describes your function? Business development, product development, researcher, management, general management, quality, investor, other. Similarly to the previous poll, you can vote in the exact same way. Please do so, and we can take a look at the results now. So 25% of the audience 25% of the audience function is business development, 50% have voted research, and 25% have voted management/general management. I will now pass over to Sebastian for the presentation.
Thank you very much, Stefan. So it was good for me actually to see who's in the audience, just to make sure that I address a little bit the specific topics. And you would all agree, I guess, that having 45 minutes is not enough to go into really a deep dive into this topic, which is very complex, which has so much really fantastic and innovative technologies to discuss and present. I give you a range, an overview, if you like, so that you might discuss this kind of technology with your colleagues, even with companies, and to really get a better grip on this technology. And if you, for example, have a need to really discuss deep technology issues or deep technology topics of microarrays, please let me know. In the end, you will have a survey form where you can add in one of the note sections some kind of feedback. So please be so kind, give me a feedback. If you want a deep dive into technology the next time, let me know. We will prepare something. So now we go ahead and I give you this overview. So microarray patch systems, and the first thing is just giving you a broad overview about what we are talking about the next 45 minutes. A quick recap of skin and which dosage forms are, let's say, trying to overcome skin, not all, just a few. Then I will give you an overview of microneedle designs, a very quick introduction to application devices, and we go into manufacturing, characterization, and development of a special kind of microneedle system. Briefly, 2 use cases, and I guess I will then be out of my breath already, and we will sum up. And at the end of this presentation, you will have the opportunity to look into a lot of literature, a lot of published literature out there to get you started with a more in-depth technology look if you want to go deeper. And of course, as mentioned, if you want to have a deep dive in technology, let me know either one-on-one. You will have contact data from me, or in a second webinar. Okay, let's jump ahead. Skin. Skin is an important barrier of the whole human body. It's not easy to overcome on purpose, actually. And when we look at skin, this is just a very rough overview. We have this distinction between epidermis and dermis, and all the hypodermis below that. And if you check the epidermis, you will see that the outer layer of the epidermis, the stratum corneum, actually is one of the main barriers to overcome when we think about transdermal or dermal drug delivery. And the viable epidermis below that has some kind of nice features as well as the dermis, because what most of us people, when we think about skin, we don't really think about the immune system. The immune system is something which is somehow inside the body, but I guess not everybody thinks about the immune system when it comes to skin. But this is basically something we have to consider, and which makes it very interesting, because when we're looking at the epidermis, there's Langerhans cells, which is part of the immune system, and all the immune competent cells in the dermis we have down there, which makes it a rich target actually for every compound which you would like to deliver to modulate or to interact with the immune system. And so the big drawback of the whole skin is we don't get the compounds there without overcoming somehow stratum corneum. And this is the big challenge we face actually when we are talking about transdermal delivery, especially when we are talking about large molecules, when we are biologics, vaccines, which are not known actually to go right over the skin. So, and to overcome this barrier actually, there are a couple of dosage forms who can do that. I just picked a few, not all, and one of those who can overcome the stratum corneum is actually classical transdermal patches, for which LTS actually is widely known for. How this works is simple. Basically, the API, the compound you would like to deliver, is dissolved in an adhesive matrix. The adhesive matrix is put on the skin surface. The API, by diffusion, by passive diffusion, is moving through this corneum into the viable epidermis and dermis region, and then it's transported via blood flow into the body. What the big challenge is, the molecule itself can only travel over the stratum corneum if it has a specific physicochemical features and physicochemical behavior, if you like. So not every molecule is able to cross stratum corneum, limiting the API range quite severely. So only a few of those are actually able to cross stratum corneum. So it's a really easy way to transport your API, but the number of APIs are limited. Of course, if you want to be sure, you're using a syringe. Injections are very common. Most of the doctors actually tell us that we use injection actually because we make sure that the dose is into the human body. And you can inject in different ways. You have the intramuscular injection, for example, going very deep, the subcutaneous injection, as well as the more modern way to inject, the intradermal injection, which is not so easily done, actually. So, of course, competent health authority, healthcare personnel can do this, and there's a special industry around that, facilitating very small needles going into the dermal region. And just to give you an idea about the stratum corneum, the outer layer you see in this picture are somehow between 10 and 25 micrometer. This is not very much. And if you think then the other layers are super thick, the viable epidermis goes something between 30 to 200 micrometer if it's a really thick epidermal region where you would never stick a needle actually. And the dermis actually is something 1,000 micrometers and upwards. So this is really targeting the dermal region. It's not so easy. Okay, so but it's the best way actually for the doctors to say the dose is in the body. Of course, you can combine this if you have, for example, a sustained release drug delivery technologies you would like to apply, because you need an insulin pump at certain intervals, you need to be dosed, or you have to have continuous pain medication to take. You can use internal or external drug pumps. This is just a quick view from the Medtronic website, which is wonderful when you would like to get more information about that. And on the left side, you see an external insulin pump, actually, with a needle sticking in the belly of this patient. And on the right side, you see an implanted drug pump, which is surgically implanted and has to be removed as well. So, of course, this overcomes the stratum corneum because it's some kind of injection, more like an infusion system. And when you compare these three drug delivery forms, and I definitely didn't go through the semi-solids, which are nice as well. So you have a mode of action in the transdermals of passive diffusion. The system by the injection and the drug pump are more or less needle-driven. So you overcome stratum corneum by punching in a needle. The onset of the transdermal patch systems is quite slow. So sustained release is more the target of the transdermal patch. In case of the injections and the drug pumps, it's quite fast because you have a liquid reservoir system where you pump in this liquid with your API into the body. Self-administration in the transdermals is not a big deal. Almost everyone knows how to stick a patch on the skin surface. In the injections, it is more complicated. I declared it as no. Of course, there are some of us who can inject this. There are these auto-injectors who help with this. Drug pumps are getting difficult as well. So I think from a self-administration point of view, transdermal patches are definitely superior to the other two. Storage conditions, transdermals typically stored at room temperature, no big deal. Typically with the small molecules, no pressure to keep it in the fridge. Injections typically have a cold chain if they are not lyophilized. And the drug pumps have also a limited shelf life. So a big disadvantage with is the API type or the limited range of APIs when it comes to transdermal patches. It's small molecules, more or less. Injection can almost all molecules inject into the body. So a big advantage here. Pain is one of the key players actually in going to alternatives from injections. Transdermal is almost, or not almost, but painless. Injections painful. I would say I'm not afraid of injections, but frankly speaking, if I could avoid it, I would. Drug pumps, yes. The first injection, yes. When it stays there, it's uncomfortable, but it's possible. And when it comes to patient compliance, pain is a big driver. This is the reason why I think patches are good and the other one are more or less poor. From a safety perspective, patches are quite safe. You apply them, you take them off if you don't want them. Injections are quite poor because if you take out your syringe, the needle itself is still there. There's a whole industry around that to protect you from getting hurt with the needle. There are safety mechanisms in place. It's not so easy. And from a usability point of view, transdermal patches are better, at least easier to use and easier to teach than to inject yourself or use the drug pumps. Right. This is a purely subjective category, but we are talking here about microneedles and how do they fit in there. And this is basically in this next slide where I say the mode of action is needle, yes, like the injections. The onset is fast. The self-administration is not a problem. Most likely, at least you have the opportunity to get rid of this cold chain you don't want to have, because we embed APIs in polymer. So you don't have a liquid you have to stabilize. You don't have to have a liquid to store. It's a solid product. So room temperature is an opportunity for you to change this. The API type is almost everything, and it's painless. And we think, and at least this is the feedback we get from the clinics, compliance is good, safety is not a problem, and it's easy to use. So a lot of arguments to go to the microneedle systems. And I'm sorry, I head over to Stefan now, for the next poll.
Thank you very much, Sebastian. Our next poll question for today is, having decided to use MAP technology, which of the following is the most important to you in selecting a microarray patch technology platform? The first option is drug loading capability. Second option is uniformity of content and dosing. The next option is clinical scale manufacturing capacity. Our fourth option is credibility of a technology provider to support you. Our fifth option for today is safety and tolerability, And the last option is I don't know. I will give the audience 10 seconds so they can answer the question. And now let's take a look at what everyone voted. So the majority of people voted uniformity of content and dosing. 34% of people voted drug loading capability. 34.3% as well voted safety and tolerability. 22.9% voted I don't know. 20% voted credibility of the technology provider to support you. I will now hand over back to Sebastian for the rest of the presentation. Thank you.
Thank you very much for answering all those questions. This helps actually giving me a good idea what you would like, and of course giving us a little bit more insight in what you as the participants actually would like to see, to know, and think. It's always difficult if it's a webinar because I can't see you. I can't raise any questions without going to a poll. So, but now again, now we continue to the microneedle designs. And I teased you with this kind of comparison with the other dosage forms. And as you see, I'm quite confident that microneedles are a very important factor in the future of drug delivery. So we look into now different designs, shapes, and types of microneedle systems because there's not only the microneedle system out there. It's a little small. You get a bigger one in the next slides. We are going to the solid removable microneedles. I give you just a brief overview. Coated microneedles, dissolving microneedles, hollow microneedles, and hydrogel forming microneedles. These are the microneedle systems we are looking at in a few slides. So we start with solid microneedles. And how does this work? It's quite easy. You have microneedles which consist of typically a metal. For example, titanium is commonly used in surgery, so you could use it also for your microneedles. So you manufacture your microneedles, and the typical microneedle can be between, I don't know, there are literature out there claiming 100, 200 micrometer in length, up to 500, 800, 900 micrometer in length. So it's a very tiny needle actually. And you have a multitude in parallel in a two-dimensional area, basically. So we typically not only use the term microneedle, we are typically use the term microarray because it's arrayed in a two-dimensional shape. So you have, I don't know, a couple of microneedles in one length and a couple of microneedles in the other length. Solid microneedles, coming back to the topic here, the solid microneedles consisting of metal are punched into the skin. So they penetrate, as you see here, stratum corneum and the viable epidermis, and they're targeting typically the dermis. They are removed, and then you place your drug on top of it. This can be a semi-solid, like a cream, a gel, or whatever, or it can also be a transdermal patch, for example, a dermal patch. And then the API moves through these tunnels, which are formed when you punch into the skin with your microneedle device. So you can imagine that a gel and these tunnels are, in addition, have a limited drug supply to the body because you have a very fast skin healing process when it comes to sustained release. It's, let's say, not the optimal way. I don't know whether we are on the same page. When I'm talking sustained release over a certain amount of time, for me, it's a couple of days. So if you want a sustained release for just a couple of hours, then I would say it's not a problem. Skin healing is turned on already, but still the channels which you build by punching in are there and can be used. So a limited drug load of the system itself might be an issue, and a short sustained release, as I said. The other issue we have is sharp-edged waste. For us, it was quite important that we wanted to circumvent this. If you pull out your microneedle design, you have still this metal microneedle in hand, and it might be possible, or it might be the case, that you punch it again accidentally into your own hand and your own finger, or whatsoever. Whatever it is still there. So you have to handle that somehow. And of course, when you apply then for sustained release your dosage form on top of the skin layer, you have a long application time, because for the time you would like to deliver your API, you have to put something on top of the skin. Okay, let's go to the next one. Via coated needles. So what's happening here is, think of the same needle that you saw in the previous slide. Take then, let's say, a polymer solution, and then you dip, for example, you dip your microneedle into this solution, and the solution actually adheres to the surface of the needle itself. You pull it out, you dry it, and you have then a coating around this needle tip. This coated needle actually is punched into the skin. The coating itself dissolves, releasing the API, and you can, after a certain amount of time, just pull out your coated needle, or the not coated needle, not anymore coated needle. You pull it out, and you throw it away. So you can imagine that the amount of API which is on the surface of a needle is limited. You can't make the needle very, very big with a very, very big coating, because you need a sharp-edged tip to punch into the skin. So you are a little bit limited compared to other dosage form or other microneedle systems. You have to check, or you have to make sure that the polymer API adhesion on the needle surface is appropriate, that the integrity of this coating is so good that it withstands the high mechanical stress it has when it's punched into the skin. Typically, this is an immediate release system, and you have sharp-edged waste because you pull out this metal needle or this plastic needle. The big advantage is it's a short application time. You punch it in, it is released most likely very fast. You pull it out, so you don't have any kind of on the skin surface any kind of medication any longer. So when it comes to the next microneedle design, it's actually hollow needles. Quite frankly speaking, it's something like an injection system. You see on the left side hand picture a reservoir layer on top of the skin, and a needle with a channel in between. And as soon as you punch it into the skin, due to the channel, you can apply a liquid reservoir, and it flows through these hollow needles into the body. And if everything is released, you pull it out, and this facilitates a high drug load, which is possible because the portion of the reservoir can be very large, actually. But you have, of course, the same problems when it's a liquid reservoir than all your injections. You can do immediate and sustained release. Nevertheless, you have sharp-edged waste again, because these hollow needles are made from metal most of the time. And yeah, depending on if you have a sustained or immediate release, you have a more short application time on the skin surface. So you are quite flexible. One before the last, the hydrogel microneedle is quite tricky. The needle itself is made out of a polymer, basically a polymer which is swellable, and it has a reservoir layer on the back. On the left-hand picture, you see it quite nicely. The tip itself is swellable, and on top of the skin lies the reservoir layer with the API. You punch it into the skin. Due to the water uptake in the skin, the polymer is swells, takes up water, and it becomes from a rigid to a more jelly consistency, and the API can move by passive diffusion through these polymer channels into the body. And if after the treatment, you just pull them out, and the hydrogel has so high cohesion that you just pull it with the back plate out. So there's without just the excipient, or not the excipient, but just the API, which is actually in the skin at the end. This gives you the reservoir gives you a high drug load, but the API still has to move in this polymer. You have limited polymer options because of those hydrogels, which are so coherent that you can pull them out, are not so easily done. You have sustained release characteristics. You have no sharp-edged waste, but typically you have a long application time on the skin surface because this systems screen for sustained release, actually. The last is the dissolvable microneedles. What's happening here is that the whole needle, like the hydrogel forming needle, is made of a polymer. But this is not a swellable polymer, but it's a dissolving polymer. The back plate on top of the skin actually has no API. It's just for delivering the needle into the body. So you punch this needle actually with this, which is completely made out of polymer excipients and API into the body. After a certain amount of time, due to the fact that this polymer is disintegrating, you can remove this back plate, as we call it, and the needle still sticks in the skin. And after a while, it disintegrates, dissolves in the fluids of the body, and releases the API quickly or in a sustained manner. The drawback is we have not this high liquid reservoir system, so the drug load is not so high like these ones. It is due to the fact that the whole needle is made of a polymer drug combination. It is medium in comparison to the other dosage forms, the other microneedle systems. You have a wide variety of polymer options. You can have immediate and sustained release depending on the polymer. You don't have no sharp-edged waste. And you have a short application time on the skin surface. Think about the following. If you want to apply a medication, you don't want maybe everybody around you should not know actually that you have medication or you take medication. Then you don't want something very big on the skin surface. And this actually is more like a temporary implant or a short-term implant by just pushing in needles into the skin and removing the rest of the medication. Okay, so as you would imagine, we at LTS actually choose the dissolvable microneedle technology, and I would like to point this out why. We don't think the other ones are not worth looking into. On the contrary, we are definitely supporting every microneedle technology out there. But we think to have a focus, it was good for us that the API load is a good compromise between the liquid reservoirs and the other systems. We have the options of having sustained and immediate release quite easily. Not that the other ones have not also some kind of options, but we thought we think this is the best compromise. We have optimal API stability due to the fact that the API is embedded in the polymer and it's not a liquid reservoir. We have a short residence time on the skin surface for treatments which should not be visible from the outside. And we have a high flexibility when it comes to needle design, length, width, number per area, and composition, because we can decide this quite fast and easy by adjusting certain manufacturing processes and compositions of the whole needle itself, not just a part of the needle. And this was actually the biggest reason to go into the dissolvable microneedles and not in the other directions, or at least have a focus in this direction. Okay, we just jump briefly in a super complex topic. This is the application devices. It's just a quick excursion. I'm so sorry that it's not getting much more attention, because it's a very interesting one. But I'm still very fast, but I think I couldn't handle this to have more than 2 slides. To give you an idea, application devices, the main question you will ask is, do I need an applicator, and which options do I have? And the question you should ask as well is the following: successful application. So successful application depends on the size of the microneedle system. Think about it: if you have a thumb large a microneedle system, just as your thumbnail as big, it's more easy than if it's a complete hand. Think about needle density. What do I mean by that? It's the number of needles per area. When it's just one needle on 1 square centimeter or 1 inch, whatever, it's easy to push into the skin. If it's 5,000 of those needles tightly packed, it's more like a solid surface pressing on top of the skin, and you don't get them in maybe by hand. So you need to check these kinds of ideas. And of course, your patient population. If you think your patient population can't handle a thumb pressing on top of a dosage form to bring it into the skin, it's you need to, let's say, standardize. And this is basically the main reason. Standardization of application. So your applicator provides you with a certain force by itself. The patient has no say in it. So you can standardize. So in general, 2 application or applicator technologies are out there, a lot more, but I categorize them in multi-use and single-use applicators. Multi-use applicators is you actually prepare some kind of your microarray in a certain way, and then you arm your applicator with this microarray. You punch it on your skin, you press a button, and the microarray is in the skin. You remove the applicator, and the next day you just arm your applicator again. The single-use applicators are just right out of the pack, typically already armed with a microarray patch, and then put on the skin, applied, and then you throw them away. So you have to answer. You ask yourself the questions: do I need the applicator, and which way do I go? Multiple use or single use? Multiple use, of course, the patient needs to somehow handle this, and the single use is quite easy, but you throw away a lot of money actually when it comes to cost of goods and cost of manufacturing. Just to give you an idea how this could look like, it's the LTS microneedle applicator. This is not because we think every microneedle system needs an applicator, but some are better off having one. And basically on the left side, you see a holding device where the microneedle is placed. You see it on the upper left picture, this black circle in the middle of the blue one. And you arm the right application device. You arm it, you put it together, you put it on your skin surface, you punch a button, and the whole disc, how we call it on the left side, is placed on the skin. It has an adhesive, so it can stay there for a certain amount of time to make sure application is secured there, and then you can take it away and the needle stays in the skin. Just to give you an impression. There's a lot of more out there. I'm sorry I can't really show you a lot of the great stuff our microneedle colleagues from other companies do. Please visit their websites. Really nice, but due to let's say rights or legal purposes, I can't just present them to you at this moment. But really nice features there. Okay, Jumping to the next point is the manufacturing of dissolvable microneedles from mixing to assembly, and gives you a little bit more insight in what LTS actually does and how we do actually our microneedle manufacturing. So the first step is always preparing the dispensing solution, meaning where the polymer and API come together. This is something where the which the microneedle is made of, if you like. So it's quite, for the researcher and for the technical guys, this is quite not very interesting typically, because when it comes to lab equipment, it's always magnetic stirrer and whatsoever, where you put your water or other solvent, your API, your polymer, some kind of plasticizers and maybe other excipients. You mix them together and you are ready. So on the left side, I put in our larger scale dispensing solution mixing equipment from our clean room area as a nice add-on. So what's happening here is actually a dispensing solution is formed. So the API, the polymer, the water. On the next slide, we dispense. Basically, we have two general principles how we can dispense. Meaning how we get this into our so-called molds. The mold is the negative of a needle, basically, and on the left side we have single tip dispensing, where we move our dispensing head, which is more like some kind of, you know it from maybe from the inkjet printing. You move over a cavity, you dispense, you move to the next cavity, you dispense until the needle is until the micro is completely dispensed. On the right side, it's a little bit different. Here, you dispense a whole area, and the dispensing solution is flowing into these cavities. It's way more faster, but you can't do a lot of fun stuff. For example, think of this single tip dispensing. Think of it that you don't dispense only one solution. You dispense 2 or 3, making it a multi-layered needle type, which can disintegrate at different times with different speeds. So there's a lot of possibilities there to enhance products. Nevertheless, of course, it takes a little bit more time when it comes to single tip dispensing. So the process as shown here is just a graphical representation. Of course, it's not just moving over each cavity, dispensing moves again. This would take too much time for the big manufacturing. So you can handle this with a lot of technology tricks. And on the left side, of course, again, pictures from our clean room areas where we have single and complete mold dispensing equipment. Just 2 examples of our dispensing units. After that, you have to remove actually the solvents. So meaning that you need to dry actually your products. And you would say, oh, Sebastian, drying, and you talk about biologics, you are not. Drying means consistent temperature over a certain time. This can be low temperature, of course. But in the end, you need to remove a little bit of water. This is not much water left there. We are talking about tiny nanoliter volumes. So we do it on a controlled condition. On the left side, you see again our clean room, one of our clean room drying ovens, which we are using there. And we can apply vacuum, for example, to actually improve these drying steps. And then in the end, it comes to demolding assembly. So you now have a dry microarray, which you can take out of the mold. You see it on the left side. And then you can either do some kind of patch system, where you have a cover on top of it for later on as an application system, where you remove this patch again and just put it by thumb pressure, or you can assemble directly by applicator and then use an application system. And this is basically the manufacturing. The next step would be characterization. This is the analytical testing. I guess the first things are quite used. You are used to these common actually chemical tests. I don't go into details there. It's very important to have some kind of structural integrity testing and see whether all your needles are present. So laser scanning microscopy comes in handy, which we use in a lot of ways. And of course, you need to make sure that your product is stable, meaning you need to actually check structural integrity here, using a compression test where you apply force and actually determine the breaking point of the needle. And if this complies to our specification, the needle is okay. We have a brief overfly of quality by design for this new dosage form. This is really just a couple of slides. We need, if we want to go into detail here, we need, I think, 3, 4, 5 hours. So actually, this is a drug device combination product, most likely for everybody else. So our authorities tell us this is the direction we should think. So you have classical from pharmaceutical quality aspect as well as the medical device ones, where needle shape, length, density is quite important, and structural integrity. The typical critical quality attributes are in place: API assay and purity, molecular weight of the polymers most likely, then a critical material attribute, and of course critical process parameters. Always when it comes to dispensing solution, mixing, storage, dispensing volume, quite important because it defines API content, time, temperature for the drug steps. And of course, when you package in a pouch, heat, sealing, time, temperature. I have 2 use cases left, and the first one actually is a recombinant vaccine animal study, where it was a proof of principle study where we wanted to compare a conventional intradermal intramuscular injection against our microneedle system. I can disclose the vaccine. It's hepatitis B surface antigen, which we used here in guinea pigs. And we had a prime boost system here, where we took samples, primed, took samples, boosted, and took samples, boosted, and did further sampling times. You see on the left side, actually, the upper side, the microarray we used in this kind of guinea pigs in the middle one. And the close-up picture actually from an applied microneedle system on top of the skin surface of a guinea pig, which has of course, no fur anywhere at the application site. And what we saw is pictured here. I'm sorry, I think it might be a little bit diffuse. Sorry for that. The threshold actually was at 10 milli-units antibody count, and it was not a problem to reach that. The graphic itself, it's always a solid column and a stripped column or a striped column. The striped column was always a test group with an adjuvant, and the solid column is without one. And you see there's no significant difference. So in this case, we didn't need actually an adjuvant to reach the same levels. What we showed in this case was you can successfully vaccinate a guinea pig, and you can do it without an adjuvant. And that this kind of macromolecular structure can be safely incorporated and manufactured with the manufacturing process you saw before in a dissolvable microneedle. So we counted this as a very big success and went on directly or into the human study with the same product basically. So we applied it at the shoulder actually, and we applied a prime boost sequence and took samples for antibody count. I can't completely disclose all the data, but I summarized them. It was safe and well tolerated. So we had mild reactions from skin surfaces like you typically have when you apply something on the skin surface. There were no severe advance. And we could actually see seroprotection at 2 of our doses we used without adjuvant. So a big success for us to see that not only in guinea pigs but also in humans we could do that. And you can imagine it took us a while actually to get ready for a human study. So we needed the whole infrastructure for a human study in place, of course, that all the authorities agreed to take this kind of dosage form, manufacture it in a certain way, and go into a human clinical study. So we were quite happy about these results. So we are at the end, and I'm really in a hurry. As to say, I think we have 10 minutes left. Just to sum up, we looked at the skin and saw that it's rich in immune competent cells. We need to overcome the skin barrier, the stratum corneum, to get the drug into the skin. How can we do that? I showed a quick overview about your microneedle systems. I showed you why LTS actually choose or focused on dissolvable microneedles. We use also other ones, but we focus really on that one. I gave you a very brief manufacturing process analytical testing summary, and a super quick overflow over the development of microneedle systems. Really, this typically takes much longer. And I showed two case studies, two in-house case studies, if you like, which we did in the past, and which show that we can use this delivery system to
Thank you very much, Sebastian. So our poll for today is, do you currently have microarray patch projects in your company? No, we're considering using the technology. MAP technology is under evaluation. MAP product is a lead program in my company. And now let's take a look at the answers. 57% of people voted no. 15% of people voted we're considering using the technology. 18% of people voted MAP technology is under evaluation. And 9% of people voted MAP product is a lead program in my company. Now I will pass over back to Sebastian.
Thank you very much. So just to stop, and from the poll, I see that you don't actually have microneedle systems in your company or in your labs or whatsoever. I would just give you an idea why you should consider microneedle systems. Think about it. It's a dosage form where patients can self-administrate an otherwise intradermal injection or a dermal injection or whatever. And this might lower the total healthcare cost for the payer, actually. No trouble to the healthcare professionals or less trouble to the healthcare professionals. Maybe you, and as a second point, have the opportunity to remove the cold chain challenges you have. Maybe more less expensive in this case. When it comes to the pain and the psychology of needle injections, you have to face that most injections have a very negative association in a lot of patients, and you might increase patient compliance and adherence to a product which they can self-apply and which doesn't hurt. And from a business development, actually think about life cycle management, extend your brand differentiate from the actual market at the moment. And nevertheless, the system itself allows a very precise and good content uniformity and adequate patient dosing, like an injection, where, as I said before, the doctors gave us the feedback. We use injections because we want to make sure the product is in the body, the dose is in the body. Think of it as a way to go there, but with less pain and more patient compliance. Okay, and now I hand over to Stefan for the Q&A.
Thank you very much, Sebastian, and thank you for that lovely presentation. Can I please remind the audience they can submit their questions via the Q&A widget? Just type them into the box and click submit. But it seems like we have a lot of questions coming in already. Our first question for today is, how critical is the application pressure for microneedle efficacy? How critical is the application pressure for microneedle efficacy?
The application pressure, so the pressure I put on my my microarray for efficacy. So when it comes to applying pressure, actually, then it's all about a right application. So if the application itself is not correct, actually you will result in a lower dose, basically. So it's important that application is suitable. Let's put it this way. There are studies published actually, which actually show that you can easily apply a microarray with a certain needle density by thumb repeatedly and consistently.
Thank you so much for that answer. Our next question for today is, what is the needle stick injury potential for MAP technologies?
The needle stick injuries, it's almost there's no actually potential for this when it comes to dissolvable microarrays, because the complete needle is disintegrating, meaning that you remove, let's say, a solid plate where you can't stick anybody, neither yourself, not somebody else, not even when you put it in the waste. So there are no sharp edges at the end after dosing. So for the, let's say, metal needles, it's a little bit different. So there's a potential of sharp-edged waste, but for the dissolvable,
Thank you so much for that answer. I'm afraid that is all the time we have for today. That just leaves me to thank Casey and Sebastian for what has been a lovely presentation, and to LTS AG for sponsoring the session. If you didn't get your question answered today, please keep in mind they can be answered at a later date. To the attendees, you receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-reviewwebinars.com. We look forward to sharing further webinars with you, so please do keep an eye out on the website just mentioned and follow us on Twitter @BRWebinars for daily updates, and join our LinkedIn group Business Review Webinars. Thank you so much once again, and I hope you have a fantastic day.